Welcome to Lecture 6.4, in which we will go over the electron transport chain and ATP synthase. This image shows the protein structures of the electron transport chains, as well as of ATP synthase. They are all called complexes because they are made up of multiple polypeptide subunits that form one large protein. Our learning objectives for this video are to describe where the electron transport chain takes place in the cell, describe its overall inputs and outputs, and understand the relationship between all the different phases of cellular respiration. We are now in the last phase of cellular respiration, the electron transport chain. The complexes that make up the electron transport chain, as well as ATP synthase, the enzyme responsible for making ATP, are all membrane proteins that are found on the cristae, the folds of the inner mitochondrial membrane. The folds in the mitochondrial membrane increase its surface area, allowing for there to be more electron transport chain proteins and more ATP to be synthesized. The ATP here represents how a lot more ATP is made at this step than in any other step. You don't need to know the number for this class, but the ballpark is generally a little over 30 ATPs made from the electron transport chain. As a whole, the electron transport chain is made of a series of 4 complexes that contain electron carriers which transfer electrons from one molecule to another. In other words, it's a chain that transports electrons. The energy that comes from the electron transfer is used to pump hydrogen ions, also called protons, across the membrane, which creates a proton gradient. In chemistry, protons are subatomic particles that have a charge of +1. Since a hydrogen ion has lost an electron and has a charge of +1, it's essentially just a proton. Again, the electron transport chain is located on the inner mitochondrial membrane. The protons are pumped from the matrix to the intermembrane space, which means that the proton gradient has a high concentration of protons in the intermembrane space and a low concentration of protons in the matrix. Overall, the electron transport chain takes an NADH and FADH2 and uses oxygen as its final electron acceptor at the end of the chain. The NADH and FADH2 are oxidized back into NAD+ and FAD, while oxygen is reduced into water. The proton gradient formed as NADH and FADH2 pass their electrons is used by ATP synthase to make ATP. Complex 1 is called NADH dehydrogenase. As the name suggests, it oxidizes NADH by removing hydrogen and 2 electrons. There are some extra details, but the electrons taken from NADH are eventually transferred to a molecule called ubiquinone, abbreviated here as Q, and this reduces it into ubiquinol, abbreviated as QH2. The QH2 will then diffuse to complex 3. What I want you to know is that during this electron transfer process, complex 1 pumps 4 protons into the intermembrane space, contributing to the proton gradient. As mentioned in the citric acid cycle video, complex 2 is also succinate dehydrogenase, the enzyme in the citric acid cycle that produces FADH2. Similar to NADH in complex 1, electrons are eventually transferred from FADH2 to Q to produce QH2, which again diffuses off to complex 3. What I want you to know is that this is the only complex that does not pump protons, and therefore contributes the least energy for ATP production. Its only contribution is the QH2 that goes to complex 3. Complex 3 is called ubiquinol cytochrome C oxidoreductase, and that's because this is where ubiquinol is oxidized and transfers its electrons to cytochrome C, which gets reduced. This is probably one of the more complicated complexes, but I want you to know is that it pumps 4 protons into the intermembrane space to contribute to the proton gradient. Complex 4 is called cytochrome C oxidase because it oxidizes cytochrome C. The electrons are now transferred to oxygen, the final electron acceptor in the electron transport chain. This produces water and also takes up 2 protons for the water. This further reduces the number of protons and also contributes to the proton gradient. Complex 4 also pumps 2 more protons into the intermembrane space. I want you to know that the formation of water and the pumping of protons both contribute to the proton gradient. I also really want you to know that oxygen being the final electron acceptor in the chain is why oxygen is so important. If there was no oxygen to receive the electrons, the cytochrome C would have nowhere to pass its electrons to, and this would eventually stop the electron transport chain from running. If the electron transport chain doesn't work, NADH and FADH2 wouldn't get oxidized into NAD+ and FAD, and all the previous phases of cellular respiration that relied on NAD+ and FAD won't be able to happen, with the exception of glycolysis. This is why high levels of NADH generally mean that there is not enough oxygen available, and why NADH is an inhibitor of the pyruvate oxidation step and the beginning of the citric acid cycle. Furthermore, the lack of electron transport also means that the proton gradient isn't produced, meaning no ATP is made. Like any concentration gradient, the proton gradient has a lot of potential energy. Protons in the intermembrane space will travel down their concentration gradient through ATP synthase. The energy released by protons going down their concentration gradient is then used by ATP synthase to create ATP. This process is called oxidative phosphorylation, named for the requirement of oxygen and the oxidation processes needed to create the proton gradient that will allow ATP synthase to phosphorylate ADP and make ATP. How exactly does ATP synthase make ATP with the proton gradient? This is a diagram representing ATP synthase, with the intermembrane space having more protons than the matrix. The black circle represents the nucleotide adenosine, and the red dots represent the phosphate groups, with ADP having 2 phosphate groups attached. ATP synthase essentially facilitates the process of combining ADP and the phosphate group into ATP. Once the ADP and phosphate group have been stabilized in ATP synthase, a proton moves into ATP synthase. As the proton moves through ATP synthase, this causes the ATP synthase to rotate. After 3 rotations, ATP is produced and leaves ATP synthase, and another ADP and phosphate group can be used to make more ATP. ATP synthase essentially works like a turbine. In this video, we learned that the electron transport chain and ATP synthase are located on the folds of the inner mitochondrial membrane, also known as a cristae. NADH and FADH2 are oxidized into NAD+ and FAD as they pass their electrons through the chain. Oxygen is the final electron acceptor and is reduced into water. As the electrons travel through the complexes 1, 3, and 4 pump protons into the intermembrane space. The resulting proton gradient is then used by ATP synthase to produce ATP, using a rotational motion. In the next video, we will learn what happens to pyruvate if there is not enough oxygen, as well as how other nutrients besides glucose can be broken down for ATP. Thanks for watching.